fortunate to recruit Keishan to the University of Chicago, where he was an outstanding house officer to the superb chief resident. In a few weeks, Keishan will be moving to Durham, North Carolina, where he will be pursuing a cardiology fellowship at Duke University. During his time at the University of Chicago, Keishan has been a model of understated excellence. His even keel and rational approach to difficult questions has been an invaluable resource for our residency program and for me in particular. Today, Keishan will be talking about cardiac-focused translational research that he has performed here throughout his residency. His grand rounds is entitled Adrenergic Signaling in the Failing Right Heart. Please join me in welcoming Keishan.
Can you guys hear me? Yep. I'm very excited to be here today to give grand rounds. For the next 25 minutes or so, I will talk to you about a clinical and research interest of mine over the past residency and membership here, and that's the syndrome of right heart failure and its relationship with adrenergic signaling. So, whether you're treating patients with obstructive sleep apnea or interstitial lung disease in pulmonary clinic, or connective tissue disease in rheumatology clinic, or morbid obesity associated with diastolic heart failure in primary care, the syndrome of right heart failure is one that is really just critical to recognize and understand. This is a brief overview of my talk today. So first, a review of RV function and the role of adrenergic signaling in heart failure. These two will then lay the groundwork for the study that I'll present to you, and this study has formed the cornerstone of my research here at the University of Chicago. Let's start with the case. Miss H is a 42-year-old female with severe pulmonary hypertension without any coexisting longer heart disease, and she's coming to the ED feeling very fatigued. In the ED, she's found to be hypotensive, tachypneic, and hypoxic. Her physical exam shows a loud V2 and a Q small sign, which is a paradoxical rise in your regular venous pressure during inspiration. Her labs show acute kidney injury, and a transthoracic echocardiogram confirms severe RV dysfunction. This is a representative photo of her echo, and you can see how the right ventricle is very dilated and compressing the left ventricle, reducing cardiac output. So Ms. H is admitted to the CCU and diuresis is initiated. However, she does need some extra inotropic support. And so the question I propose to you is this. What is the optimal inotrope to use in RV failure? And what is the evidence behind this? So this is an excerpt from a text by Dr. William Harvey from back in 1628. And in this text, Dr. Harvey makes the first known description of the pulmonary circulation, and also within the context of the complete circulation. In this quote and throughout the text, he also wonders what the function of the right ventricle is. So he says here, and why was nature reduced to the necessity of adding another ventricle for the sole purpose of nourishing the lungs? So clearly Dr. Harvey is not a pulmonologist, but he does note that there are inherent differences between the left and right ventricle. Now this slide will highlight some of these differences. Let's start with embryology. So the right ventricle arises from laryngeal mesoderm, and this is different from the left side of the heart, which comes from the early heart tube. Also, the septum is an RV structure. When you look at the right ventricle structurally, it actually has the same stroke volume as the left ventricle. It only requires a quarter of its stroke work. And this is because the right ventricle is part of a low-resistance pulmonary circuit. The geometry of the right ventricle is also quite complex. And this is a photo of the right ventricle at both end-diastole and end-systole. And you can see that when it contracts, much of its contractility is actually derived from longitudinal shortening. This is different from the left ventricle, in which circumferential shortening is the main contributor to contractility. Finally, perfusion. The right ventricle receives its coronary blood flow in approximately equal amounts both systole and diastole. And this is in contrast to the left ventricle, in which most of it is actually during diastole. So I've shown you several important differences between the left and the right ventricle. And although there's still more to learn about it, we are gaining a deeper appreciation of its prognostic significance. And RV function is one of the most important prognosticators of pulmonary hypertension. Having said that, I still want to acknowledge that there is still a very complicated relationship between the left and right ventricles. So let's go back to Miss H. What does coming into the hospital with the diagnosis of right heart failure mean for her? This is a single center study of 205 consecutive hospitalizations and 90 patients with pulmonary arterial hypertension. The leading cause of admission was right heart failure, and you can see that these patients are very sick. So this is the freedom from death or transplantation post. But let's take a look at in-hospital mortality. It was 14% compared to the cited 3 to 5% for left heart failure admissions. Furthermore, if these patients were put on inotropes, their mortality in the hospital went up a lot. So this raises a question of cause and effect. Sure, maybe these patients were just really sick and were going to die anyway. But perhaps the inotropes led to increased mortality, as it does in left heart failure sometimes. The authors of the study also looked at inotrope selection, and you can see that there's really a mishmash of inotropes. Dopamine was the most prevalent one used, but there's no clear way of giving inotropes in these patients. And this is at a single center, and there's even a much less consensus nationally. I want you to take away one point from this slide. So this is a schematic diagram of the right of the heart, and so in normal. Sorry about that. So in the normal heart, you can see the normal right ventricle with the thin wall and its corresponding pulmonary artery. When you have RV hypertrophy, you can see how the walls are thicker here, and it's actually fairly well concentrated. So that's one point to take away. The cardiac output is normal to mildly decreased. When this progresses to RV failure, you can see how the walls are getting thinned out, and the RV is actually much more dilated. And this is characterized by increased endothelial cell proliferation and apoptosis in the pulmonary artery. RV failure has a severe decrease in cardiac output, and I want you to take that point away from this slide. Let's shift gears for a second. So, as you know, there are a lot of trials in cardiology, but really only a few of them have had game-changing benefit, and this is one of them. So, this is the beta blocker trials from the 1990s, and patients in this trial in particular had LV systolic dysfunction, and they were randomized to either a beta blocker or placebo. And you can see that the patients on provedolol actually did much better. They had a 38% increased probability of event-free survival. And now, beta blocker and beta adrenergic signaling actually form some of the cornerstones of therapy in left ventricular systolic dysfunction. But what does this mean for the right side of the heart? Well, we don't quite know yet, but we do have some data about adrenergic signaling in pulmonary arterial hypertension. This was a study done by Dr. Rich in the 90s as well, and it looks at his patients with pulmonary arterial hypertension. And so we first grouped them into tertiles by their random plasma norepinephrine level, as you can see here. He then estimated their survival based on severity of pulmonary hypertension. And you can see clearly here that the patients that have the highest plasma norepinephrine levels also have the worst survival. So maybe there is a connection between the adrenergic signaling system and RV failure, as we know that there is in the left side of the heart. Having looked at all this data, let's re-examine our question for MIS-H in this context. So, what is the optimal inotrope in RV failure, and what's the evidence behind this? The rest of my talk, I'm going to take you through my investigation of this question over the past 4 years, and I'd like to frame this talk in the following way. So first, we'll talk about models of RV hypertrophy and RV failure. We'll also talk about optimal inotrope selection, and we'll use what we've learned to find a potential therapeutic target for RV failure. So let's start with the RV hypertrophy models. We used rats as our models, and shown here are cross-sections of their hearts. There's a control, an adaptive, and 2 maladaptive models. You can see in control that the LV and RV are shown here, and the RV has a thin wall. Looking at the PA banding model, now we call it adaptive because it has RV hypertrophy, but it's compensated. There's no coexisting RV failure. The PA banding model was created by ligating the pulmonary artery, and this induces RV hypertrophy. This mimics pulmonary stenosis. The 2 maladaptive models now. So chronic hypoxia insusion is the next one. These rats were raised in a chronic hypoxic chamber, and they were given Sugen. Sugen is a VEGF inhibitor. And we call it maladaptive because the RV hypertrophy then So monoproline is an inflammatory substance. When it's injected, it causes a widespread inflammatory reaction, including lung toxicity and pulmonary artery endothelial cell dysfunction. And so we have our models. Let's see how they did. This looks at both treadmill distance and cardiac output of our rats. And you can see that in all of our experimental models, both treadmill distance and cardiac output were reduced. More so in the maladaptive models with RV failure, though. To study the contractility of these hearts, we use what's called a Langendorf heart apparatus. And it lets us look at the heart in isolation. So the way we do this is to first anesthetize rats and isolate the heart. We then attach the heart by the aortic grooves to this tube. And there's a perfusate here that contains both calcium and nutrients. We let the perfusate run through the ventricle, and it will also retrogradely perfuse the coronary arteries. This allows the heart to continue beating on its own for several hours after we've isolated it. Now, like I said, we're looking at the heart in isolation, and the advantage of this is that we take away both preload and afterload. We get a focus on the heart's properties itself. We're also able to attach electrodes to the left ventricle and the right ventricle and measure systolic pressure. And because we don't have preload and afterload, systolic pressure is now a surrogate for contractility. Let's look at some of our results. So this shows RV contractile reserve. Contractile reserve is the ability to augment upon the pre-existing contractility. And so these are our 4 models. The y-axis is the relative right ventricular pressure. The x-axis is a logarithmic scale of increasing doses of medications. And these gray areas here are the ranges at which these drugs are physiologically active. So let's look at control and see if you have an example. So you can see that dobutamine will increase contractility by almost 3-fold in the control rat. I want you to take away just 2 points from this slide. The first one is that contractile reserve is reduced in all of our experimental models. You can see that the peak right ventricular systolic pressure is less than that in the controls. And the second point to take away is that dobutamine is doing a better job in improving contractility than dopamine does. You can see how it's higher in all of our graphs here. We also took a look at isoproterenol. So isoproterenol is an inotrope that will stimulate beta-1 receptors. And you can see similarly, the contractile reserve is reduced in our rats with RV failure. And finally, we looked at phenylephrine, which is not an inotrope, it's an alpha-1 agonist. But because it does get used quite often for treating RV failure, we wanted to see its contractility effects. And there's minimal improvement in contractility in the rats with RV failure. We also wanted to see what this means in vivo, and to do that, we constructed pressure volume loops. So shown here are control rats and the rats with the maladaptive RV hypertrophy. The y-axis shows RV pressure, and the x-axis shows RV volume. And these black lines here are the baseline pressure volume loops. When we added dobutamine or dopamine, we constructed new pressure volume loops to show the effects. And we can calculate stroke volume using the end-diastolic volume and the end-systolic volume. And we then calculated the increased stroke volume. So you can see that here for control rats. And this is what we found when we looked at the rats with RV failure. There was dobutamine did a significantly better job in improving stroke volume than dopamine did in RV failure. Again, consistent with what we found prior to this. So, showing you a lot of data, but I want you to take away so far that 1, contractile reserve is reduced in all of our models of RV hypertrophy, and 2, dobutamine is a superior inotrope in rescuing the RV while it's failing. To further investigate what our findings so far means, we took a look at the adrenergic receptor expression in the right ventricle. And so this is the beta-1 adrenergic receptor and its mRNA expression. You can see that in all of our experimental models, beta-1 mRNA expression is reduced. So take a look at the TV screen. I think it's a little bit easier to see there. This is the post-translation protein expression of beta-1 receptors in the right ventricle. So this is immunostaining. So each row here represents a different experimental model, and each column represents staining for a different cellular structure. So green is staining for the beta-1 receptor, Red is dystrophin, which will stand for cellular membranes. Blue stands for Dept, or the nucleated cells. And the last column is a composite of the prior three. So two things to take away from this slide. First, direct your attention to the dystrophin column. And you can see just how severe the RV hypertrophy is in our experimental models by looking at the size of the myocytes, comparing it to the size of the myocytes in the control. The next thing to take away is from the beta 1 column. And you can see that the amount of green goes down as you go down the column. What does this mean? Well, it means that the density of beta 1 receptors is decreased in RV hypertrophy and even more so in RV failure. Okay, consistent with what we found so far. We also wanted to see what this means in humans. So these are age and sex matched humans with controls and those with pulmonary arterial hypertension. And just keep in mind that the colors are switched here. So now red represents beta 1 receptors and green represents dystrophin. And all you need to do is look down each column and see that the staining for red is decreased. So the expression of beta 1 receptors is decreased in humans with pulmonary arterial hypertension as well. So dopamine affects beta 1 receptors, but it also affects dopaminergic receptors. There are 5 subtypes of dopaminergic receptors, but they've actually never been studied in the right ventricle before. From what we know in the left side of the heart, D1 is the primary contributor to inotropy, and D3 and D5 are as well, but to a lesser extent. When we looked at mRNA expression of the D1 receptor, we found, interestingly, that in the adaptive model, it's relatively well preserved, but it's downregulated when you have RV failure. And we found similar patterns for both D3 and D5. Finally, we looked at alpha-1 receptors in the RV. So alpha-1 is the target for phenylephrine, and alpha-1A in particular is a primary target for phenylephrine in the heart. And you can see, again, significant downregulation of all alpha-1A receptor mRNA in the right ventricle. Let's just shift gears for a minute and look at the effects of right heart failure on LV contractility. So what's going on the other side of the heart in the setting of RV failure? Again, these are our Langendorf models, and the y-axis now represents LV systolic pressure, and the x-axis is the same as before. One point to take away, so let's take a look at the control first, actually. You can see that there's almost a 2-fold increase in contractility with dobutamine. But the one thing to take away from this slide is that in the setting of RV failure, that is in maladaptive models, you can see that dobutamine still does a better job than dopamine in improving contractility. So let's just take a step back and summarize what I've shown you so far. So these are the cross sections of the rat's hearts. So the normal RV and normal LV. I want you to imagine that each of these hearts represents contractility. So there's beta 1, dopaminergic, and alpha-1 receptors. I've shown you that in the adaptive RVH model, there is downregulation of these receptors in the right ventricle, especially the beta 1. In the maladaptive model that has RV failure, this downregulation is more severe, and it also involves the left side of the heart. The question is, can we rescue this model and improve contractility? So we explored this question by looking at the beta 1 adrenergic receptor and its downstream signaling cascade. So catecholamine stimulates the beta 1 receptor and then activates this pathway in which cyclic AMP is produced and contractility is increased. Now there's a molecule called GRK2, and it's part of a negative feedback loop in which it can block the actions of this beta 1 receptor. So cyclic AMP is no longer made, and the increased contractility isn't there anymore. So again, GRK2 is part of a negative feedback loop for this beta 1 receptor. And actually, a group in Rochester, New York, took a look at the effects of blocking GRK2. So they used mice that have LV failure, and they gave some of the mice a small molecular compound called gallium. Gallium will block GRK2, and then they looked at the effects on LV contractility. And you can see over the next few weeks, these rats had an improvement in the left ventricular contractility. What does this mean in our models with RV hypertrophy? augment contractility. And you can see the controls and the rats with RV failure. The point to take away from this graph is that dobutamine does a better job in improving cyclic AMP in both the controls and those rats with RV failure. The next thing we looked at was GRK2 activity. So remember, GRK2 is an inhibitory molecule that will reduce contractility. When we give catecholamines to the controls, the level of GRK2 goes up as expected. When we give catecholamines to the rats with RV failure, there's really no difference in the level of GRK2 activity, meaning that it's already at its maximal level. So this is what we proposed. What if we can use a molecule like galanine and block the effects of GRK2 in the right ventricle, thereby letting more cyclic AMP be produced and increasing contractility in these rats with RV failure? So that's what we did. We injected galanine in our experimental models and took a look at cardiac output and treadmill distance. So shown here are our adaptive models with PA bending and the maladaptive models that were monocotyl. And you can see that there was a significant increase in cardiac output in our RV failure model and a significant increase in treadmill distance in our adaptive model. And these were trends. So again, I've shown you a lot of data, but if you take away one thing from this slide, I want you to remember this. From this up, additive remodeling and receptor downregulation contribute to RV failure. I've also shown you that RV contractile reserve is reduced in all of our models with RV hypertrophy. If the RV hypertrophy progresses to RV failure, left ventricular contractility is also reduced. I've also shown you that dobutamine is a superior inotrope in improving contractility in the setting of RV failure. And finally, the blockade of GRK2 may actually augment contractility and is potential therapeutic in RV failure. So this project's been very meaningful to me. It's grown my appreciation of cardiology, physiology, and the scientific process. But I've also been really lucky to make some really great friendships and relationships in the lab. So I'd like to thank people in the Archer Lab, especially Lynn Howe, who's been a great mentor for me and taught me a lot of the skills. She's got a lot of patience, and thank you for that. Young Hu Feng, who helped with creating the models and a lot of the invasive measurements. Peter Toth was really great in terms of teaching me immunostaining and capturing a lot of these images that you've seen. And John Ryan, one of our recent fellows from the cardiology program, helped a lot with the echo measurements. Of course, Dr. Archer, who has, I'd like to thank for letting me work in his lab, first of all, and also just being a mentor for me throughout the years. I'd like to thank my co-chiefs. It's been a really great year, and I really thank you for your support. Dr. McConville has been a great program director, and thank you very much, Dr. McConville, for all your help over the past year. Dr. Oakes and Dr. Weiss, thank you for letting me give this grant rounds. And again, my wife, Anita, and my parents, Uma and Cherish, who are here today, and without their support, I would not have been able to do any of this. So thank you very much.
Wow, one wonders how you even had time to be a chief resident. It's an amazing amount of work and beautifully presented. Let me ask just an introductory question in terms of, I presume you use primarily male mice as a model, or not?
I don't know.
I mean rats.
Yes.
Male.
Yeah.
Male rats. So is there any evidence that there's different responsiveness to catecholamines between men and women, both clinically and in the lab?
That's a really good question. We actually didn't look at gender specific, but I think there is a lot of data coming out about gender differences in autonomic cases, and I think that would be interesting for both left and right heart failure. I don't know the answer to that, though.
Yes. Hi.
Hi.
Marty.
So, as you can imagine, I'm very well versed in Archer's lab and research, and you've done an amazing job presenting it all. I think the question that I have for you is that the actual model itself is isolating
think that's a,
Stimulates, like, because the take-home message could be, you should use fennel in RV failure, which isn't the take-home message. And so I want to try to figure out how you can make this model more similar to the take-home experience.
I think that's a really good question. I'm actually glad you brought that up because that really is not the point of the talk to say that one is better to use in RV failure. The question is, which is better in improving contractility? And as we know, phenylephrine has other really good benefits in terms of increasing perfusion of the right ventricle. And unfortunately, that's not the point of the study, but we can say from the study that if we need inotropic support, that dobutamine may improve contractility of the right ventricle. It's pretty much, I think, and then potentially therapeutic parts from that pathway. But I think that's what we can take away from this study.
Dr. Fink?
In the use of dobutamine, you have done mainly animal work. Is there any human work on this? And what is the degree of improvement with this drug? How long does it last? Can you have any rough idea there?
So another really good question. So the question was, is there any parallel work being done in humans, and how long does the blockade of GRK2 last, right? So there unfortunately is not a lot going on with humans right now that I know of. Actually, Dr. Rich's study from the 90s was the most closely related to this. There is another study that looked at rats and the ability of their RV failure, of their RV to recover from failure, but there's not a lot about this. GRK2, however, is a molecule that is, I think, being increasingly looked at in left heart failure. There's actually, so galenine was the drug that we used, but there are actually many potential compounds that can block GRK2. And actually, I think that's a good future direction for this project, is looking at those other molecules. We're not quite there yet in terms of translating this to humans by any means yet.
Well, these two presentations were absolutely beautiful and certainly bode well for the future chief residents that we have in our program. I also would like to personally thank John DeCombo, who is program director, really nurtured all the chief residents and the ones that we're going to hear about in the coming weeks, hear their presentations. So I thank everybody and applaud